Okay, now we are going to talk about mitosis and cytokinesis and what happens in each of the parts of mitosis and cytokinesis. As you recall, in the cell cycle, you have interphase, which consists of gap 1, synthesis, and gap 2, when the cell is doing its job. If it's an eye cell, it is seeing. If it's found in your diaphragm, it's helping you breathe. So during this time, the cell is doing its job. In addition, it has some other things. In gap 1, it is growing, so it becomes full size and replicating their organelles. In synthesis, it's copying the DNA. And finally, in gap 2, it's finishing its job and doing a little additional growth. So during interphase, the point of interphase is for the cell to do its job. But in addition, it also can prepare the cell to divide during the S phase. If it gets a signal to start S phase, it will start copying the DNA. So mitosis and cytokinesis ends up, because you're copying the DNA in synthesis, producing two identical cells with the same genetics. All right. Now there's 4 phases to mitosis. The first phase is called prophase, and that's when the chromosomes will supercoil, like I showed you in the last video, condense into these little packages, and then the spindle fibers will form from the centrosome area and the centrioles. Okay, centrioles, centrosome area, spits out, it reorganizes all of the cytoskeleton so that they make these fibers that eventually will attach to each side of the sister chromatids and pull them apart. Now let's talk about why the chromosomes need to supercoil. If you've ever seen balls of yarn maybe loose, maybe some of you have parents who knit or crochet, if you wanted to separate 2 skeins of yarn, would it be easier to separate them if they were loose and all easy to tangle, or would it be better if you kind of rolled them into a ball or somehow condensed them? So it's the same thing with your chromosomes. Rather than having them loose and easy to get tangled, what your body does is supercoil them, like your parents might supercoil or roll the yarn into a ball so that it's easier to separate them. Okay, so this happens during prophase, and prophase is the very first phase over here of mitosis. Mitosis means we're dividing the DNA. All right, the second phase of mitosis is called metaphase. Meta. So meta means kind of in the middle. And what happens here is all the chromatids and their sisters attached at the centromere line up single file in the center of the cell. Let's go back for a second. There's 3 things that happen in prophase that you need to keep track of. 1 is that the chromosomes supercoil. 2 is that the centrioles and the centrosome area reorganize the cytoskeleton to make these fibers, these spindle fibers, which we'll see in a minute what they do. And the 3rd thing is that the nucleus or the nuclear membrane will disappear. That's so that the whole cell, we have room in the whole cell to line up our chromosomes and separate them. Now in metaphase, all the chromosomes line up in the center and the spindle fibers grow until at least every single sister chromatid is attached on either side to one of the spindle fibers. So there's some spindle fibers that are not attached to any of the chromosomes, and they will help make the cell longer, elongate it so that it's easier to split it. But the most important thing that happens in metaphase is that all 46 chromosomes line up in the center, and each sister chromatid is attached on either end to a separate spindle fiber. This is the second phase over here, metaphase. Okay, let's look at the third phase, which is anaphase. In anaphase, the sister chromatids that were attached at the centromere are now being yanked apart from each other. And remember, it's kind of like yarn. So I know they were set up like an X here, but it's kind of soft like yarn. And so when you yank on, or these spindle fibers are pulling on this sister chromatid, it turns kind of like that, like a V shape. I hope that makes sense. We can talk about it in class if you have a question. So each of the spindle fibers pull apart the sister chromatids. So now this side of the cell will have a full set of all 46 chromosomes, and this side will have a full set of all 46 chromosomes. Meanwhile, the cell's getting a little bit longer and the spindle fibers are shortening to pull the chromosomes apart from each other. In anaphase, the two sister chromatids are pulled apart from each other. In the last phase of the mitosis part is telophase. In telophase is pretty much the opposite of prophase. Now that all of the chromosomes have been separated from their sisters and pulled to opposite sides of the cell, the spindle fibers begin to shorten and disappear, and the cytoskeleton reappears to do its job, which is to create the, to hold the cell out and to make some pathways through the cell, etc. This nuclear envelope, or the nuclear membranes, reappear, and although you can't see it in this picture, the sister chromatids will now unravel and become loose in the cell, more like a bowl of spaghetti, so that the DNA can now be turned into proteins. Okay, this is the last phase of mitosis, called telophase. Finally, in order to completely separate the two cells, the last part of this cell division is cytokinesis. You can see cyto means cell and kinesis means to move. And what happens is some fibers that are kind of like movement, or let's see, they're not muscle, but they're actin fibers which help move things, will draw pinch the two cells apart until they're separate cells. You can see it pinching right here in this in this microscope view of cells separating. And of course, now we've got our nuclear envelope intact. We've got the nucleus intact. You can see the DNA has uncoiled and is loose like a little bowl of spaghetti. The centrosomes now are have stopped making the spindle fibers, and we've got two cells that are identical. This is cytokinesis. Now, plant cells and animal cells do cytokinesis differently. Here's those actin fibers that are pinching in the center of this cell until it makes two cells. Here you can see a little more clearly down here, until you have two cells. And in a plant cell, what happens is that little vesicles, remember vesicles are small circles of membrane that have something inside. These vesicles have cellulose, little sections of cellulose in them, and they all migrate to the center of a plant cell, because a plant cell has a cell wall around it and it cannot pinch in like an animal cell. And once all of these little vesicles have lined up in the center, the cellulose kind of joins together until it forms this cell plate, and the membranes that were around the vesicle fuse together to make the membrane on either side of this new budding cell wall. So you can see that in plant cells, they kind of build a cell wall. You can see all the vesicles here, and pretty soon all the vesicles are going to fuse together so that the cellulose makes a straight line or a wall, and on either side the membrane will extend, coming off of the opening up the vesicles. You know, the vesicles are surrounded by cell membrane. Now that cell membrane is gonna, or not cell membrane, but membrane is going to be part of the new cell membrane. Thank you for listening.